Welcome back, everybody. So just to open our first medical session of the day, this session is entitled MPN 301. I'm not entirely sure what happened to 201, but we're jumping straight to 301. First up, I'd like to welcome back to the stage Martin Ellis, who you will remember gave us a really good grounding yesterday morning, and I'm sure he's got a few of the slides that we had to cut him off with yesterday. But please welcome back to the stage Martin Ellis. Thank you.
So, good morning, everybody. It's me again. And I think that my task for this morning is to set the stage perhaps for Stefan, for the second speaker who will speak about the implication of these mutations. So what I'm going to try and do is just take you through some of the mutations that we deal with in the MPNs. And the topic was chosen, the title of the topic was chosen, Know Your ABCs. I've added here, dealing with these multiple muddling mutations to try and keep the alliteration going. So what I'd like to do is start off by perhaps dealing with the names of some of these mutations. Where do they originate? Sometimes it helps us understand what they mean. It's not always important, but sometimes it's interesting. I'd like to talk a little bit about the functional aspects of these gene mutations. What are they actually doing in the cell, to give you some insights into that. And then I will talk very briefly about their prognostic importance in general, because I have a feeling that the next speaker will speak more to the actual clinical implications of these mutations in patients. So this is a scary slide, which is from a very well-known publication from the New England Journal of Medicine a number of years ago. And what it does is outline the mutated genes that you can find in populations of patients. You have the percentages of patients over here in clinical trials and in observational studies in whom particular mutations were found. And you can see the vast number of mutations that have been described in patients in various clinical trials. And again, as said, the percentage of patients in whom these genes are found, these are obviously patients with myeloid malignancies, myeloproliferative neoplasms. And one can see over here the most frequently mutated gene, as we spoke about yesterday morning, is, as expected, perhaps the Jak2 mutation, which occurs here in upward of 60% of patients. We have the CALR gene, which we spoke about to some extent yesterday, coming in at number 2. And then we have a variety of other genes whose names are perhaps always appellations are a little more obscure. TET2, ASXL1, DMT3A. We'll talk about some of these and what they mean, but you can see this list goes on and on and on and on and on. It's almost unending. The color coding over here is telling us both the structural abnormality that is found in the gene and to some extent also what this actually may do to the function of the gene, if it increases the functionality of the gene or decreases the functionality of the gene, and how that may affect the particular cell and the development and function of those cells. So if we think about a particular cell in cancer in general and in our diseases, the MPNs in particular, how many mutations may we find in a particular cell? So it turns out that in hematological malignancies, we actually are in quite a good position. We have the luxury of having malignancies in which the cells contain, on average, depending on the malignancy, somewhere in the region of about maybe between 10 and 20 gene mutations per malignant cell. If we think about diseases like lung cancer, pancreatic cancer, there can be up to 70 genes mutated in each malignant cell in those solid organ tumors, which perhaps gives us some idea as to why some of those diseases are much more difficult to treat and are much less treatable, certainly less curable than some of the hematological malignancies. We mentioned chronic myeloid leukemia yesterday as one of the myeloid malignancies that is not a classical MPN. It's the Philadelphia positive myeloproliferative neoplasm. And we mentioned there that there is one gene that is mutated that actually causes the disease, or one chromosomal abnormality leading to one gene, a new gene formation, a novel gene, the BCR-ABL translocation. So that's one gene, one disease model. The MPNs have many more gene mutations in them, and the more complex and the more difficult to deal with the MPN, the more gene mutations there are. This is sort of intuitive, but it happens to be true as well. So there are more gene mutations in myelofibrosis patients than there are, for example, in PV or in ET. That would be in descending order. And again, by comparison to some other malignant diseases like cancer of the pancreas or lung, there are relatively few gene mutations in these diseases. And so this is just a flavor of what we're dealing with in the number of mutations across the spectrum of patients that we may encounter. As we mentioned yesterday, the most important gene mutations are those that are either primary in the disease or causative to some extent in the disease. We call those the driver mutations. And then those that come along afterwards, which can modify the course of the disease, would be called the passenger gene mutations. Interestingly, research has shown that if you profile a patient's gene mutation pattern at the time of diagnosis, you have a fairly good snapshot of what the gene mutation profile is going to look like over time. So patients who have certain gene mutations usually tend to keep those gene mutations, and that's what defines their prognosis to a large extent, as we'll see. And they generally do not accumulate more gene mutations over time. Sometimes they do, and when that does happen, that's usually an indication that transformation is afoot, usually transformation to an acute myeloid leukemia, and sometimes transformation to myelofibrosis. So when we look at the various gene mutations that are present in patients, they are usually stable over time. So it's not the kind of testing that we would do on a repeat basis to see whether a patient has accumulated more of these gene mutations that appear on the x-axis over here. So let's just look at some of the more common names, and just for curiosity's sake, have a look at where they come from. So the Jak2 mutation is a mutation in the Jak gene. If we say that's a Jak2 mutation, that means probably there's a Jak1 and a Jak3. Turns out that there are, of course, and they're also important, but we focus on Jak. And Jak over here stands for Janus kinase, and the Janus kinase gene was named when it was discovered for the god of beginnings, with the two heads, one looking forward and one looking back. The name was given because, as we showed in the cell membrane yesterday, this Jak mutation involves a mutation on the tail of the receptor for various cytokines, those chemicals that speak to the cells, the soluble chemicals in the plasma of patients that speak to the cells. So this receptor is receiving a message from outside of the membrane and transducing or sending the message in towards the nucleus of the cell, as I showed you yesterday. So this is sort of looking backwards and forwards, or it's a bidirectional mechanism that occurs here. This receptor gets a message from outside and transfers it inside. There were cynics at the time who thought, well, this is a kinase gene. The kinase implies an enzyme activity. Any word that we use that ends with the letters ASEAs implies that we're talking about an enzyme. So this is an enzyme that adds those phosphorus groups onto the receptor, as I showed you, those little P's, which stood for phosphorus, which activate the enzymatic activity, the protein activity of the Jak proteins. So they said, well, this is a Jak kinase. It's very interesting. You can call it a Janus kinase. But in actual fact, it's just another kinase, which would be the other abbreviation for which the Jak stands. So that was a cynical approach, saying, well, this is just another enzyme. It's not going to turn out to be that important in cells. So for a change, the cynics were wrong. And as we all know since then, the Jak has been critical in the MPN. So it's not just another kinase, but it is perhaps the kinase that is involved in the MPNs. And that's where the name Jak comes from. Calreticulin, being the second commonest mutation that we have in the MPNs, as I told you, the name is a composite of calcium and the reticuloplasmic part of the internal part of the cell, one of the organs in the cell. So that's a composite which implies that the protein is found in the endoplasmic reticulum. It's part of the internal workings of the cell, the machinery of the cell that has to do with protein folding and protein production. And it's a very important binder of calcium. So that's why that name was chosen for that gene. The third driver mutation is the Mpl Mapple Mutation in the Mapple gene. And that stands for myeloproliferative leukemia. This was a name that was given to this gene when it was discovered originally in mice many years ago. And from here it gets a little bit more esoteric. Now we're into the passenger mutations. These are mutations that are not considered causative in the myeloproliferative neoplasms, but they certainly do affect prognosis. And one of the most important prognosis-affecting genes, gene mutations that we have is in a gene called ASXL1. And ASXL stands for additional sex chromosomes-like, ASXL. And the nomenclature here relates to development of organisms, humans and non-vertebrate organisms. The name is obscure, the reference is obscure, but that's what it stands for. And a lot of these genes, and some of them we'll talk about now, have to do with developmental processes and the monitoring and the modulation of organ development and the control of organ development. So very basic biologic functions all the way back to embryology, to the developing embryos of vertebrates and non-vertebrates alike. So it's a name that doesn't have any particular meaning as far as myeloproliferative neoplasms are concerned, but it's a gene that is frequently mutated and has important prognostic implications. Likewise, EZH, that's another gene that is frequently mutated, is one of the passenger mutations. And this stands for enhancer of zest homolog, which again refers to an embryological process. Who exactly gave it this name, I do not know, but that's what it stands for. That's the implication of these three letters, that's where it comes from. Now here's one that makes a little more sense. It's just as esoteric, SRSF. This is an important mutation as well. And here it stands for serine and arginine-rich splicing factor. The S and the R, the SAR, serine and arginine, are two amino acids. And this implies that this is a gene that has an effect in certain processing of the protein production of proteins that are rich in these two amino acids, the serine and the arginine. Again, very esoteric, not really relevant, certainly not clinically or relevant to patients, but biologically very important. And at least this abbreviation has some relevance to what is actually going on. So it's an enzyme or a gene that's involved in the splicing or the cutting of various fragments of DNA and RNA in order to produce proteins correctly. And if you have a mutation in this gene, it will affect cell function. So this has some relevance. The next is the Tec mutation, which I mentioned yesterday as one of the perhaps earlier mutations, even occurring prior to the JAK2 in certain cases of myeloproliferative neoplasms. And again, here, the letters stand for 10 to 11. It's not the kind of thing that you would intuitively know, but this implies a chromosomal translocation, so a break between chromosomes 10 and 11, and a piece of chromosome 10 jumping onto chromosome 11 and creating a new gene which has certain function and causes abnormalities in the cell. And somebody, interestingly, decided to call this TET, which stands for 10 to 11. This was probably the researcher in the lab who was using some kind of abbreviation to make his or her life easier. And finally, a gene that's not really common in myeloproliferative diseases, but is common in other hematologic diseases. I just thought I'd bring it to show you how variable the names can be. This is called VAV. This was a mutation in a gene that was described in a laboratory in Boston by an Israeli researcher who was working there, and VAV is the sixth letter of the Hebrew language, and this woman discovered this mutation in the sixth run of a particular electrophoresis study that she was doing, and she called it number 6, which again, unless you knew the reference to that, would make no sense to anybody at all. So that is just a very brief list of some of the more important and more obscure and more unusual names that we have for these gene mutations. The message is that you can't understand really what the mutation is telling you or what the mutation is doing in most cases. And they have reference to either biological, embryological, and sometimes to idiosyncratic laboratory workers' penchants for giving interesting abbreviations to their findings. What we can do, though, is try and understand how some of these genes affect cell function. And what I'd like to show you in this cartoon over here, going back to our schematic that we had yesterday, if we look at this bilipid membrane over here represented by this dark line, similar to the membrane that I showed you yesterday. So the top part of the slide would be the extracellular portion. This is the cell being bathed in its liquid, the plasma in the body. And below this line, we have what's going on inside the cell. So this is the intracellular portion of the slide. We're familiar, I think, with the structure over here. This is the transmembrane receptor, which receives a signal from a soluble molecule, it can be a hormone or another chemical, some of which are called cytokines, some of which are called integrins, and that chemical will bind specifically to this receptor structure over here, cause change in the conformation of the receptor, and that will start the conversation or the process of signal transduction between molecules within the cell, ultimately in the context of myeloproliferative neoplasms, giving the nucleus an instruction to replicate and for cells to proliferate, and in some cases to proliferate in an abnormal manner or in a dysfunctional manner, certainly in a numerically abnormal manner in the myeloproliferative neoplasms. So we have certain genes that are involved with signal transduction. Some of them, again, importantly, we have discussed. The Jak2 is over here, the Mapple is over here. Other genes that are important in other myeloid diseases. Some are important in acute myeloid leukemia, like this FLT3 gene. Some are important in mastocytosis, which we mentioned in passing yesterday, another chronic myeloid malignancy. And these would be a group of gene abnormalities that are involved in signal transduction. Now, if we move into the cell, we will see over here a group of genes, perhaps start up over here, that are important in the regulation of the formation of RNA. I think you're aware that within the chromosomes we have DNA. That DNA is transcribed into RNA, so it moves from one nucleotide structure to another, and it's the RNA that is translated into protein, and it's the proteins that carry out the functions of the cell. So the sequence would be DNA, RNA, protein, in terms of causing cellular functions to occur or allowing cellular functions to occur. So it's the signal transduction that takes us into the nucleus. The message is passed from outside the cell through the cell membrane via its receptor over here. The talk between various molecules, which we call signal transduction in the cytoplasm, ultimately ending up in the nucleus. These are chromosomes that are shown over here. And then that results in transcription, which means activation of the DNA, and its a transfer into RNA, and finally protein. So if we look at the DNA, we've seen how DNA is affected by signal transduction. There's some more examples over here that are less relevant to myeloproliferative neoplasms that we're talking about. NPM1 is a very important gene in acute myeloid leukemia. Then we can move on to RNA regulation, and the formation of RNA is regulated by a number of enzymes. Again, each enzyme has its own gene which encodes for it, and here are some of the abbreviations which we have seen: SF3B1, SRSF2, which I mentioned before, and some others over here, again, having these long abbreviations, each of which means something. And these will be a series or a collection of genes that are important in the regulation of RNA development and function, and mutations in these genes may cause abnormalities in the RNA part of the story over here, which ultimately will cause cells to become more malignant, less regulated, and allow disease to progress or disease to transform, again, as we mentioned yesterday, from a more chronic phase disease to a more acute phase disease. Again, there are a number of diseases, a number of genes that are responsible for the regulation of transcription of the actual mechanism of DNA multiplication. We have some of them over here. This is a protein called TP53, which is made by a gene called P53, which is perhaps known to some of you. This is a very important gene in cancer development in general and in myeloid malignancies in particular. And this is a very unfortunate gene because mutations in this gene are usually very, very violent mutations that can cause rapid transformation to diseases like acute leukemia. And these are mutations that are usually not present at the time of diagnosis and would be the type of mutation that a patient can acquire and would explain why the disease has transformed, and usually transformed quite aggressively, to a much more aggressive form of myeloid leukemia. There are other examples over here of genes that can affect transcription relation as well. And finally, once the protein has been produced within the cell, there are very important processes that occur after the genetic events have occurred, or on top of the genetic events, therefore the name epigenetic, meaning on top of the genetic, and this usually refers to changes in the protein that occur because of the addition or subtraction of various substances, chemical substances, which can affect the way that proteins work. And these are very important genes in our myeloid malignancies, the MPNs, because mutations in these particular genes that affect the epigenetic regulation, things to do with methionine and cysteine, cytosine and cysteine metabolism as relates to proteins, have very, very important effects on the way that the proteins are folded, the way the proteins are structured, and therefore the way the proteins function. And ultimately, it's protein function that is important in the cell. The genes are only there to create the proteins. So the gene abnormalities are important. Once the protein has been synthesized, if further changes occur to the protein structure, as would happen in abnormalities of these genes over here, then those proteins can still malfunction. Even though the genes creating a certain protein may have been normal, once the protein has been created, yet another series of abnormalities can occur in those proteins. And these are called epigenetic abnormalities. And you'll recognize perhaps some of these abbreviations, one or two which I mentioned earlier, particularly the ASXL1, which confers a very adverse prognosis in diseases such as myelofibrosis. So again, just to look at these important non-driver mutations, so the mutations in addition to the JAK2 and calreticulin, we have the list over here of 7 or so important mutations in primary myelofibrosis. I mentioned yesterday that cytogenetic abnormalities are moving quite quickly into the field of Polycythemia Vera, and here are some of the important genes that would be associated with adverse prognosis in Polycythemia Vera, and even essential thrombocythemia now is starting to have genes recognized in this disease which can confer an adverse prognosis. Just very quickly to finish off, yesterday I went through a number of the prognostic scoring systems that we have, and we started with the ancient one from about 10 years ago, the International Prognostic Scoring System, which required eye contact with the patient to ask very simple questions like, how old are you and how do you feel? And now we're moving on to what is called over here the genetically inspired prognostic scoring system, which theoretically you could do for your patient even without ever meeting him. And if you have a look over here, there's absolutely nothing in this scoring system which is clinical at all. This is purely based on the chromosomal abnormalities which may be present in a patient's cells, the driver mutations. And we said yesterday that having the absence of a calreticulin mutation is a bad thing. Having a calreticulin mutation type 1 is actually a good thing, relatively speaking. And then we have over here the passenger mutations which confer high risk on the patients. And you're familiar already with these Kaplan-Meier survival curves over here, which can, again, in the same way that we saw with the clinical prognostic scoring, but in a much more detailed way and a much more precise way, provide us with long-term prognostic information about our patients at the time of diagnosis. So we've moved from a clinical prognostic scoring all the way to a genetic prognostic scoring with the important ones, the DIPSS and the DIPSS plus and the MIP70 and so forth in between, which are probably the ones that are more commonly used in daily practice because they combine both the clinical and the genetic and genomic abnormalities. But here's an example of one of the more modern prognostic scoring systems which really takes into account these driver and the passenger mutations that I've tried to tell you something about this morning. Thank you very much.
Thank